everyone, my name is Jenna Benke and I am the Nature and Conservation Intern at the Conococheague Institute. Today we'll be doing a freshwater monitoring at our Wetlands Trail Pond using the Lamont Low-Cost Water Monitoring Kit. Freshwater monitoring is important because throughout the U.S. each state has different regulations for different levels of sediments and pollutions. Here at CI, we want to make sure our water is safe for the frogs, turtles, and birds, and even humans that walk the trail. Today we will be collecting a water sample from the wetlands pond and performing the test for the following. The first is coliform bacteria. Fecal coliform bacteria is naturally present in our digestive tracts, but the presence of this bacteria in water is dangerous and indicates fecal contamination. Now, the water in the wetlands pond is not consumed as drinking water, but it is important to test for this bacteria, as water flows into the Welsh Run, which then connects to larger bodies of water in the Potomac watershed. For this test, we want to see low numbers of coliform bacteria. Next is dissolved oxygen. Dissolved oxygen, which is important to the health of aquatic animals and ecosystems. Natural waters with high levels of dissolved oxygen are rare and provide stable environments for a diverse population of aquatic species. Dissolved oxygen can be affected by water temperature, season, and human-induced changes. For example, agricultural runoff can increase biochemical levels in water, such as nitrates, which can affect dissolved oxygen levels through algal blooms. For this test, we want to see high levels of dissolved oxygen, as low results can indicate hypoxic environments. Next is biochemical oxygen demand. This measures the quantity of dissolved oxygen used by bacteria as they break down organic wastes. In slow-moving water, such as our wetlands pond, available dissolved oxygen is frequently consumed by bacteria, depriving aquatic organisms from the dissolved oxygen they need to live. For this test, we want to see lower biochemical oxygen demand levels because that indicates that dissolved oxygen is available for aquatic organisms and not being taken up by bacteria. For nitrates and phosphates, this test will indicate how many parts per million of nitrates and phosphates are in the wetlands pond. Nitrates and phosphates usually enter water sources through agricultural runoff or animal manure. So based on our surroundings here at CI, we can hypothesize that the wetlands ponds has high levels of nitrates and phosphates. When nitrate and phosphate levels are high, this can lead to eutrophication, which is the process that involves an excess growth of algae, blocking sunlight and decreasing photosynthesis rates, which also contributes to low levels of dissolved oxygen. For pH, this test measures the acidic or basic quality of the water samples. The pH scale ranges from 0, which is very acidic, to 14, or very basic or alkaline, with 7 being neutral. Fresh water typically falls between 6.5 and 8.2, and maintaining this level is crucial to the survival of aquatic species. For temperature, this test we will be using the provided temperature sticks which have been adhered onto the container kit. Along with nitrates, phosphates, biochemical oxygen demand, and pH, temperature also affects dissolved oxygen levels in the water and the rate of photosynthesis. Temperature can be affected by a wide variety of environmental factors, including time of day, season, geographic location, and of course the infrastructure that surrounds the area, such as agricultural fields. Finally, we will be measuring the turbidity of the water using a Secchi disc, which has been adhered to the bottom of our test kit container. Turbidity is another word for water clarity and is important to test because it is an overall pollution indicator. When turbidity is low, this can disrupt natural processes in the pond such as light penetration and photosynthesis. Turbidity is visually measured by lowering the disc into the water just until it is not visible anymore. We will then estimate the approximate distance from the surface of the water to the Secchi disc. On April 14th, I collected a sample and these are the results. The temperature was 14 degrees Celsius, nitrates were about 40 parts per million, which is not good, phosphates 2 parts per million, which is fair, pH 7.5, which is good, dissolved oxygen was 8 parts per million, also good, turbidity was 20 Jackson turbidity units or 78% clarity. So our coliform was definitely on the positive side, so we do have fecal bacteria in the wetlands pond. And our dissolved oxygen was between a 6 and an 8 parts per million. So our results were as we expected. There's a lot of nitrates from agricultural runoff, but thankfully we do have some life here. We have some frogs, we have some turtles, we have lots of snails. This project and internship was very beneficial to me as I would like to continue my career in water and soil science at Chesapeake Bay Foundation. I am recently graduated from Wilson College in Chambersburg, Pennsylvania with a dual major of animal studies with a concentration of environmental science and environmental sustainability. My paid internship at CI was made possible by a PHMC grant and generous donations made by Jan and Lee Davis and the Stafford family.